taught you that diagram. So first let me open the cell biology part. Where is biomolecules here? Cell biology, cell cycle, cell division, yes, cell structure, then cilia and flagella. Look, this is the diagram which you need to draw. So you need to draw only what? This diagram, just this one. You need to not
Okay,
draw these one. You need not to draw these one, right? You have to draw like this one, this part, right? This basal body, then plasma membrane, then this one, microtubule. Then you have to draw just a circle, right? Then let me draw it in front of you. If suppose that if somebody asks you that draw the well-labeled diagram of cilia and flagella, so what you need to draw? Look, flagella, it's very simple. Flagella, when we study the flagella, we recall the chlamydomonas. Chlamydomonas have two flagella. So just draw the chlamydomonas structure, draw two flagella. So they are the flagella, right? And cilia, you will get the best example is paramecium. So paramecium hold the cilia, right? It bear the cilia. Then you can draw the diagram like this. So this is the flagella and cilia, right? First, right? So this is
okay, sir.
flagella and this is cilia. So first you have drawn this diagram. Then I have told you there is only difference, that is the length of the cilia and flagella and number of the cilia and flagella. Flagella can be 2 to 8 maximum or 4, generally 4. Cilia can be numerous, right? And cilia are very small in the length, and I have told the length that is 200 micrometer. Micron is the length of flagella, but cilia can be smaller one, approximately 39 micron, right? Then what you have to draw? You have to draw a circle. Then I have already told you there is a 9+2 structure which is found in the cilia and flagella. So there will be 9 doublet. 1, 2, 3, 4, 5, 6, 7, 8, 9. And at the center you have to draw 2, right? And then you have to write it down that this is the membrane or plasma membrane, right? They are the 9+2 arrangement. And what are they? They are called microtubules. So basically they are made up of microtubule doublet, right? And then the central part is called axoneme. Yes, remember axoneme. The same
Yes,
thing is
sir.
over there. Look, the same thing is given over here. Then you have to make it simple. That's it, and that's all. Look, hardly it have taken 1 minute to draw the diagram, right? You draw this diagram and
Okay,
your
sir.
purpose is solved. That's it. Got it? So that
Yes, sir. Got
yeah,
it.
that will be the it will make your answer perfect. Any more doubt?
No, sir.
Okay, great. So come to again, we were in the photosynthesis part. Yes, this, yes, we were here. So now come to the rest of the experiment. Now this is very important. These all experiments are very important when you talk about the your NEET examination, and even in your school examination, the 1 marker question for they are very important. Joseph Priestley. So Joseph Priestley was a scientist and who said that he hypothesized. I will dictate it. He said that plant convert phlogiston into dephlogiston, and phlogiston he given a term. Phlogiston means foul air, means polluted air. So he said in the nutshell, he said that the foul plant have the capability to convert foul, means impure air to into pure air, means phlogiston into dephlogiston. And he did his experiment in the mint plant, right? So you need to remember what is the phlogiston, what is dephlogiston. Then Lavoisier came. So Lavoisier said that this phlogiston is carbon dioxide and dephlogiston is oxygen, right? After that, a scientist, one more scientist came. His name was the von Mayer. Von Mayer given the a theory that plant can convert solar energy into chemical energy by the process of photosynthesis, right? Then there are 3 scientists, Ruben, Hasid, and Kamen, right? They said that the O2 comes from water, not from the carbon dioxide. And O18, he used O18 to prove his experiment and to do experiment. Then Kelvin, we will study Kelvin. That is Kelvin cycle is there. Kelvin showed the pathway of carbon dioxide fixation with the help of C14, and the process of carbon fixation was explained by Kelvin, and he got the Nobel Prize for that, right? And then HN6, T4 pathway. Leave that. We'll study all that, right? So what you have to note down? You have to note down the Joseph Priestley, Lavoisier, von Meyer, Rubin, and Kaplan. Note down. I will dictate the experiment of Joseph Priestley. Put the Joseph Priestley hypothesized Joseph Priestley hypothesized that foul air or phlogiston produced during burning of candle or animal respiration could be converted into pure air or dephlogiston by plant. Full stop. He did experiment with mint plant, right? Then next point, Lavoisier explained that phlogiston is CO2 and dephlogiston is O2. Next point. Next, you note it down like this way, the way they are written over here. Let me know when you note it down. Done? Completed?
Yes, sir.
Okay. So this was all about the previous experiment, and now the next thing, where does photosynthesis takes place? Here first you need to understand. Look, I have already explained you regarding the chloroplast. Remember, we have already discussed the structure of chloroplast in cell biology part. Structure or the the unit of life, right? In cell biology part, we have already discussed in detail, and I said on that time that you will need it in the photosynthesis part. So look, chloroplast have dual membrane, outer and inner membrane, and then there are some coin-like structure. These coin-like structure are called thylakoid. When these coin pile up in a big structure, these are called they are called grana, right? So granum is singular term and grana is a plural term, right? And the space inside the thylakoid means these coin, they are called lumen. That space is called lumen. and these remains interconnected with each other with the uh, these stroma lamella. These interconnections are called stroma lamella, and inside there is a gel-like structure. This gray color structure which appear over here, that is called stroma, right? Okay. Then one very important thing which you need to understand. Focus over here. In case if you didn't get, I will explain it again. Before going into that, I want to ask you one thing. So these are the leaves. Leaf have some special kind of the cell which are responsible for photosynthesis. We call them mesophyll cells, right? And in mesophyll cells, the chloroplast are arranged in the peripheral part. I repeat, the leaves have multiple layer, right? Anatomy of plant we have already done, and inside the leaves there are special kind of the cell. We call them mesophyll cell. They are meant for photosynthesis. And in mesophyll cell, the chloroplast are arranged in the peripheral part of the cell. Why? Tell me. Ilam, Shidika, Martin, all of you. Alia. Tell me, I have already explained to you. That's bad if you don't tell me back. Why chloroplast are arranged peripherally in the mesophyll cell? That's not fair.
So that they get sunlight at bigger at a larger surface area, like
Very good. Very good. Very good. Excellent. Optimum utilization of sunlight, right? Suppose that the leaves are there at the sunrise time, the sunlight is here. They will get the light for the photosynthesis. At the mid of the day, the sunlight will fall from here. They will do the photosynthesis at the time of the sunset at this point. So everywhere, from wherever the light is coming, irrespective of the direction, they will get the light and they will perform the photosynthesis. Excellent, right? So I want this kind of the answer from all of you, right? Remember, whenever I say to you, okay, so photosynthesis takes place in two parts: light reaction and dark reaction, right? So we can divide, technically we divide photosynthesis in two parts: light reaction and dark reaction. This is written over here. I will give you the time to write it down. What is the light reaction? Breakdown of water molecule is called light reaction. Why? Because this reaction takes place in presence of light. That's why we call it light reaction. This is light dependent. Where it takes place? This light reaction takes place in these thylakoid. Coin-like structure. Thylakoid means these, when number of thylakoid are arranged one upon another, these thylakoids are known as grana, right? So this is the grana, right? This is a grana. So inside grana, this is called thylakoid. Thylakoid. So light reaction takes place in grana. Then what is the dark reaction? So dark reaction is carbon dioxide fixation. Plant take carbon dioxide in the form of gas, but they fix it. What does it mean, fixation? Means they convert carbon dioxide in some chemical, right? This is light independent reaction, right? So means this reaction do not require light. That's why we call it dark reaction. It doesn't mean that it take place in dark. It do not require light. That's why we call it dark reaction, right? This reaction takes place in stroma. So tell me, dark reaction will go on, or it will take place daytime only, night time only, day and night both, none of the above. Quick, don't take too much time. Dark reaction will take place only in daytime, only in night time, day and night both, fourth, none of above. Elam, what should be the answer? Martin? Shridika? Yeah.
Sir.
Shridika? Alia?
So what's the question?
Question is that dark reaction will take place. First option in daytime only. Second option, night time only. Third option, day and night both. Fourth option, none of above. Come on, guys. I have already told you the answer.
Maybe day and night, because it
Well,
takes the product, it uses the product of light reaction. So as soon as the light reaction is completed, it will take. Yeah, okay.
don't be that much technical. Be simple. Try to
Yeah.
think simply.
Yes.
Look, it
So it
do
will
not
take night both, because it can take place in presence and without the presence of sunlight.
exactly. I said it do not require light. It doesn't mean that it will not take place in presence of light. It do not require light. It will take at daytime as well as take night time. Light reaction, it require light, so it will take place only in daytime, not in night time. Clear? Right?
Yes.
Note down first this part. Then I will go to above at the top of PPT. Note down this one. I will let me light it down. Look, takes place only at daytime. Takes place at day and night both. As it do not require light. Let me know when you complete it.
Then, sir.
Okay. Fine.
Done, sir.
Great. Now try to understand what I am telling you, and you have to make this diagram as well right now. First, I want 100% focus of yours. When I will complete it, first you have to draw this diagram, which I have drawn with the help of my own pen. Right, this one, this light green color part. Then you have to draw this diagram. This is complete explanation of the light reaction and direct reaction. Try to understand. Look. Chloroplast structure we have already understood in detail. So this is the outer membrane, this is the inner membrane, right? This gel-like part is called stroma. These are called structure called grana. I have already told you in granum means single structure, coin-like structure, coin pile up of coin. It's called granum. And when we include all these granum, we call it grana, right? Because granum is the singular word and grana is a plural word. Then one more important. Look, individually, I have taken out thylakoid. Just take out the thylakoid. In thylakoid, try to understand those part which are attached with another thylakoid, right? Those portions which are attached with another thylakoid. So this portion is attached with another thylakoid, right? This part is called appressed part. Appressed part. That's part which is in contact with another part. That is called appressed part. So these part where both are in contact with each other called appressed part, and those part which are free from, that means free part. This outer part, this outer surface which is free, that is called non-appressed part, right? So in appressed part means those part which are in contact with each other, right? So in appressed part there are a type of photosystem. I will explain to you what are the photosystem. So photosystem are basically a special type of group of chlorophyll molecule, right? So inside chloroplast, there are a special kind of the molecule which give green color to the plant and responsible for photosynthesis. We call them chlorophyll. So in appressed part, there are photosystem 2, right? This photosystem 2 is a group of chlorophyll molecule. There are collection of 250 to 400 chlorophyll molecule and they are found, right? They are responsible for breakdown of water molecule, right? So breakdown of water molecule takes place in where? Appressed part, where? In photosystem 2, right? Then in non-appressed part, there are another kind of a special kind of chlorophyll molecule, group of molecule. They are called photosystem 1, PS1, right? PS2 means photosystem 2, PS1 photosystem 1. They are found in non-appressed part, right? They are not responsible for breakdown of water, right? It means the photolysis of water. Breakdown of water molecule means PS2, photolysis of water. That is H2O breakdown into 2 hydrogen ions and 2 electron plus O2. This is called light reaction or Hill reaction. This takes place only daytime in presence of light and it takes place in PS2. Did you get it or not? Tell me. Everybody.
Got
From
it,
everybody.
sir.
Got
Got
it?
it, sir.
Okay.
Yes, sir.
Fine. That's why I want you to draw this diagram right now, right? But this is very clearly, right? So the next one is that dark reaction. So this was light reaction. Why? Because breakdown of water molecule means photolysis of water molecule takes place only in daytime. Now come to the stroma, right? Dark reaction, which do not require light, right? Dark reaction takes place in stroma. In stroma, the carbon dioxide comes and carbon dioxide fixation takes place. The process of carbon dioxide fixation takes place with the help of a cycle. The process of carbon dioxide fixation, I have already explained. Carbon dioxide fixation means taking in the carbon dioxide gas and converting it into some chemical, right? Means fixing it, right? Then carbon dioxide fixation takes place in Kelvin cycle. This part is called CO2 fixation and it takes place in dark. So that's why it do not require light. That's why it is called dark reaction and they take place in day and light both, right? Day and night both, right? This is called. So this part take place stroma, right? So light reaction, breakdown of water molecule. Dark reaction, that is the fixation of carbon dioxide. Both we have understood. And light reaction takes place in grana, dark reaction in stroma, right? The structure of thylakoid I have already explained you. I will explain you further what are the PS1 and PS2. Before going into that, first go through with the diagram, go through with this schematic diagram means this process. Let me know if there is any doubt. If you don't have any doubt, you have to note it down, copy paste in your notebook as it is right now. Tell me if you have any doubt. Ilam, Shidika, Martin, Alia, all of you.
No, sir.
No, sir.
Martin, today you are not speaking. Everything clear? Getting everything?
Yes, sir. Yes, sir. Yeah.
Great. So please note it down this one because you won't found this complete diagram in any book. I have not copied from any book. I have just explained it to make you understand. Right. First, you have to draw this. Don't draw all the granum. Right. Just draw single granum. Single granum. Right. Then make this arrow, photolysis of water, and thylakoid. Take out the one arrow from thylakoid and draw the PS1, PS2. Because in next slide I will tell you what is the PS1, PS2. Please note down right now.
Done, sir.
Done? Oh, okay. When I ask you to draw, explain the dark reaction or light reaction, then you have to explain this one, right? When I ask you light reaction, then you have to explain like photolysis of water, right? At least you have to draw one thylakoid. Let me know rest of the people when you do it. Okay, so Shidika, you have done, right?
Yes, sir.
So tell me, I have a question for you.
Yes.
There are 7 color in light, right?
In?
There are 7 color in light
Yes, sir.
that you know, right? With your. So
Yes, sir.
out of these 7 color, in which individual color the photosynthesis will be maximum, and in which individual color the photosynthesis will be least?
Um.
This question is for all of you. Out of these 7 colors, in which color the photosynthesis will be maximum, in which it least? This question is very basic and very old question of medical examination. It has been asked multiple times. Anyone of you?
Photosynthesis will be most in green and the
Okay.
least in um blue.
Yeah, Least in blue. Okay. What about you, Elam? Alia and Martin. Yes, Elam, are you saying something?
Yes,
Where it will be maximum and where the least?
so maximum will be green only and
Maximum will be green and
I'm not sure, sir.
I'm not sure. Alia and Martin, do you have any view or not? Look, guys. Suppose that I have wore this blue shirt. It means it appears blue because it reflects back blue light. Out of 7 color, it absorbs rest of the 6 color, but it is reflecting back blue color. That's why it appears blue to you and me. Plant appear green because they absorb rest of the 6 color, but they reflect back green color. If they will reflect back all green color, then how it's possible that photosynthesis will take place maximum in green color? The photosynthesis takes place least in green color because plant reflect back completely green color. This was the first tense. Got it? The logic behind it? The photosynthesis maximum takes place in red color, then in blue color. Please note down this statement, please. The photosynthesis takes place maximum in red color light, then in blue color light. Full stop. It takes place least in green color light. It takes place least in green color light. Right? Got it? Fine. This is structure we have already understood that this is palisade layer. This is mesophyll part, and mesophyll is made up of palisade layer. This vertical layer, palisade layer, and there is spongy parenchyma. Right. So please now note it down some point below this diagram. First, I want to tell you what is the PAR. Any idea what is the PAR? PAR. Photosynthetically active radiation. PAR stand for. Let me note it down. PAR, that stand for photosynthetically active radiation. Please note down the part of a spectrum used in photosynthesis, the part of a spectrum used in photosynthesis have wavelength between 400 to 700 nanometer. It is called photosynthetically active radiation. Full stop. Leaves appear green. Leaves appear green because chlorophyll. Leaves appear green because chlorophyll do not absorb green light. Rather, it reflect back. And the most effective I have already told you. Next point. The chloroplast, next point, the chloroplast align themselves, chloroplast align themselves in vertical position, chloroplast align themselves in vertical position along the lateral wall. Lateral wall in high light intensity. In high light intensity. And along the tangential wall, along the tangential wall in moderate light. Full stop. Change in position. Change in position help chloroplast to achieve optimum light for photosynthesis. Full stop. A mesophyll cell may have three-handed chloroplast. Next point. The matrix or stroma contain DNA, RNA. The matrix or stroma contain DNA, RNA, ribosome, enzyme, etc. Next point. The thylakoid contain photosystem second. Thylakoid contain photosystem second in oppressed part. Where water molecule breaks down. Full stop. Photosystem 1 in bracket PS1. Photosystem 1 is found in non-appressed part photosystem 1 is found in non-appressed part. Full stop. So this was all about, right? One more statement. Light reaction takes place at grana, and dark reaction takes place at stroma. Clear? Is that clear?
Yes, sir.
Fine. Give me 1 second. One more thing I want to make you understand. This is for your making you to understand only, right? Then I will tell you what is the photosystem 1, what is the photosystem 2. Before going into, and that is the only, like, what is light reaction? PS1, PS2. Yes, that is there in your NCRT. Try to understand what I am going to deliver you. I want your focus over here. So I told you that this is the grana, right? Oppressed part and non-oppressed part, you have understood already. This is a grana, means single. This is grana. And in granum, we already came to know that the breakdown of water molecule will take place here, because photosystem 2 is present over here. This is the PS 2, right? PS 2 is present over here. So here what will happen? The breakdown of water molecule take place in presence of light, right? So light will fall over here, and due to falling of light, this water molecule will break. Due to breakdown of the water molecule, 2 hydrogen ion will be formed, right? Plus 2 electron will be formed, right? Is that clear? And what else will be formed? Oxygen molecule will be formed, O2 will be formed. Because of these 2 electrons, try to understand, right? Because of these electron, the electron transport chain will be produced, right? And this complete process takes place in presence of light, so we call it light reaction, right? So this is the light reaction. Is it clear? Light reaction. Is that clear?
Yes, sir.
Look, when this light reaction will take place, because of these electrons, I want your 100% focus over it. You won't get these things in book, right? Because of this hydrogen ion and because of these, basically these 2 electrons, right? Because of these electrons, leave the 2 or 3 or 4 electron, that is a different story. Because of this electron, a cycle will go inside, where a cycle, electron cycle will go inside these thylakoid, right? Because of these electron, a cycle with this is called electron transport system, ETS, right? Where this ETS will take place? In grana, right? Inside grana, the electron transport system will take place, right? Because of this electron transport system, what will happen? 2 things will be produced. That is a ATP, right? ATP will be produced, right? And finally, these electron will be taken place. So along with the ATP, NADPH will be produced. So what is the outcome of light reaction? These 2 things are outcome of light reaction. I will write it down. These 2 things are outcome of light reaction. Is that clear to everyone?
Yes, sir.
Okay. Shridika, are you getting it? Martin?
Yes, sir.
Okay. So what is NADPH? Can you tell me? Can anybody tell me what is the NADPH? NADPH. What is NADPH? What is this? It's bird, it's buffalo, deer, devil. What is this? Look, NADPH is also an energy molecule like ATP, but NADPH is a bigger energy molecule, right? 1 NADPH is equal to 3 ATP. Got it or not? 3 ATP. It means NADPH is also energy molecule. Is that clear?
Yes,
So-
sir.
Remember all these things, right? Now this complete part was light reaction. Now this NADPH and ATP will be utilized in where? In dark reaction. In dark reaction, right? So what is the dark reaction? Dark reaction takes place where? In stroma, right? What will happen? The carbon dioxide will come from atmosphere. CO2 is coming from environment, atmosphere. This CO2 is getting fixed in a cycle, right? This is called Kelvin cycle or C3 cycle. In Kelvin cycle or C3 cycle, this carbon dioxide will be fixed and finally it is converted into a product called glucose molecule. Here the ATP, the ATP and NADP, NAATP and NADPH will be consumed, right? It will be utilized. So what have been whatever produced in light reaction that have been consumed in dark reaction. If light reaction will stop, then dark reaction will also stop. Is that clear?
Yes, sir.
Sir.
Right. Okay, great. So ATP and NADPH will be consumed. Then what will be produced? ATP will be consumed and it is broken down into ATP. NADPH will be consumed and it will be converted into NADP plus or NAD plus. Yeah. So this is the complete process of photosynthesis. Tell me if it's clear. Better you draw it. This is a schematic diagram of complete photosynthesis. This complete reaction is technically called photosynthesis. Actually. And if you have any doubt, you can ask. The heading will be photosystem.
Done, sir.
Done. Everybody done? Note it down.
1 second, sir.
Yeah, sure.
Done, sir.
Now come to this press part. I have already told you that these part which are in contact with each other, they are called press part, and that part bear photosynthesis 2, PS2. In short, we call it PS2, photosystem 2, where the water molecule breaks down, and these are called PS1, which are found in non-press part. Right. These exposed part are called non-aqueous part. Then look, what is the photosystem? Photosystem is the collection of a special kind of molecule. Basically, this is chlorophyll molecule. They trap the light and they collect the energy, and that energy is utilized for the breakdown of water molecule. Look, photosystem 1 is also known as P700, right? Because it captures the light of 700 wavelength, right? Look, there is a center. At the center, there is a reaction center. The center part or core is made up of reaction center. The reaction center is basically made up of this primary electron acceptor, which will accept the electron, and reaction center that is made up of chlorophyll A molecule, and that chlorophyll A molecule is called P700, right? And rest of the molecule, other than this reaction center chlorophyll A molecule, rest of the molecules are known as the antenna pigment. These antenna pigment are made up of carotenoid and chlorophyll B molecule. These antenna pigment, this antenna part, they absorb small frequency of the light, small amount of the light, and finally they deliver their light or deliver their energy to the reaction center. When reaction center get the energy, it breaks down the water molecule, and because of breakdown of the water molecule breaks down, an electron come out. These electrons are accepted or received by primary electron acceptor. So PS1, they are found in non-aqueous part. They have more chlorophyll A molecule. Chlorophyll A and they are made up of chlorophyll B and carotenoid molecule are found in less amount, right? Photocenter is made up of dimer, means two molecule of chlorophyll A. They are called P700. They takes part in both cyclic and non-cyclic electron transport chain, right? And they are responsible for photophosphorylation. Photophosphorylation is a technical term. The meaning of photophosphorylation means formation of ATP. Is that clear?
Yes, sir.
So please don't draw complete diagram. Put the heading PS1 and P700 and just draw a reaction center, 2 or 3 antenna molecule and primary electron acceptor. Write down PS1, P700. Done?
Done, sir.
Okay. Then Martin, note it down.
Done, sir.
Great. Then come to the photosystem second. Photosystem first and second, they are the 2-marker question, and if it have not been yet covered in your school examination, this is very important. They will definitely ask you that explain what is the PS1 or PS2, right? Then come to the PS2. PS2 is found in appress part. They have the same structure. The structure, if they ask you the diagram will be same for PS1 as well as for same PS2, right? There will be both the photosystem will have, they have the reaction center, the primary electron acceptor, and there will be antenna molecule. That's it, right? This is the same diagram applicable for both of them. PS2, it is found in the appress part of thylakoid. They have chlorophyll A, chlorophyll B, and carotenoid molecule. The chlorophyll A and chlorophyll B are present in equal amount. Here you will see that more chlorophyll A molecule, chlorophyll B and carotenoid are in the less amount, right? Here, actually this question have been asked in NEET, that's why I have underlined it multiple times, right? Then the PS2, it will have chlorophyll A and B in equal amount, right? Carotenoid are higher in PS1, right? Photocenter is a dimer of chlorophyll A molecule, but here the dimer of chlorophyll A molecule is P680. So here the reaction center have P700. That's why chlorophyll PS1 is called P700 as well, right? Why PS2 is called P680? Because the chlorophyll A molecule is P680. It received the wavelength of 680 nanometer, right? That's why it is called, it's a dimer of P680, right? Here, one more thing is found, that is the oxygen evolving complex, OEC, which is responsible for evolution of oxygen. Oxygen evolving complex is also found in PS2, which is made up of Mn double positive, calcium double positive, means ions of manganese, calcium, chloride, right? And oxygen evolving complex is responsible for breakdown of water molecule. Here, the 2 molecule of water molecule breakdown, formation of 4 hydrogen and 1 oxygen, and 4 electron takes place. This complete process called photolysis. Photolysis takes place in PS2 because of OEC, oxygen evolving complex. Please note it down, right? Note down all the points. And note down this reaction. This is the actual reaction of photolysis of water. Done?
Yes, sir.
Everybody done? Should we move ahead?
Yes, sir.
Okay. This was the light reaction. Now come to the electron transport chain. So electron transport chain or ETC is of 2 types, cyclic and non-cyclic. Non-cyclic is that where the electron starts its journey and then it do not come back, right? And in cyclic, the electron starts its journey and then it comes back in the same point, right? Look, that you have to, here you have to cram it, right? There is nothing, these all things, they all are, these all component are found in the membrane of this one, thylakoid. Look, electron transport chain, the non-cyclic, it is called Hill reaction. This is also called non-cyclic photophosphorylation. Non-cyclic because electron do not come back. Finally, electron is taken by NADP plus and it converted to NADPH. And first, what happened? Photolysis of water takes place. That we have already understood. When water molecule breakdown and photolysis of water takes place, then this these 4 electrons are released. These 4 electrons are taken by or received by P680. We know that P680 or photosystem second part, its core of photosystem second. This PS second take the 4 electron. These 4 electron get the energy when they are received by the P680 because it have already taken the energy from the light, right? Electron jump. Electron reaches to electron acceptor. Then it is received by the in membrane. It is received by pheophytin, right? I have encircled pheophytin. After pheophytin, this electron reaches to cytochrome B6F complex, right? This is called cytochrome B6F complex. This one, right? Then cytochrome B6F complex, when it reaches to over here, formation of ATP takes place. Then electron reaches to plastocunine, then cytochrome B6F, then electron reaches to PC, plastocyanin. I have written over here that pheo means pheophytin, PQ means plastocunine. Look, pheo means pheophytin, PQ means plastocunine. Then it reaches to cyto B6F. It means cytochrome B6F complex, right? Then electron reaches to PC. Everything which you need to note down in your notes, that in your notebook, that I have encircled, right? Rest of the thing you can leave. When electron passes through plastocunine cytochrome B6F complex, formation of ATP takes place from ADP. Then finally, plastocyanin give electron to P700, means photosystem 1. Again, photosystem 1 take the energy from outside, means from light, and give this energy to electron. Electron again get energy and jump. When electron jump, they get excited. They reaches to A naught. From A naught to Fx, right? Fx to FD. FD means ferredoxin, right? Fx or Fs means FeS complex. That means ferrous and sulfide complex, right? Then it reaches to ferredoxin. From ferredoxin, these electrons are taken by 2 NADP plus. When 2 NADP plus take electron, it convert into NADPH, right? This complete. So finally, the electron originated from water molecule breakdown, and it is finally taken by NADP plus, right? That is the final acceptor of electron, right? Due to this one, electron do not comes back. So that's why it is called non-cyclic photophosphorylation, right? Photophosphorylation means formation of ATP. I have written over here. Here I have written 1 GTP is equal to 1 ATP, 1 NADPH is equal to 3 ATP, 1 FADH is equal to 2 ATP, right? So this, when you get the question that please explain or draw the well-level non-cyclic photophosphorylation, then you have to draw this diagram on. Tell me if you have any doubt. Here you need to not cram it. There is no other way. Ask me if you have any doubt, any one of you. Here there is no other method. That the thing which is required, I have already explained. Here you have to like cram it. Shudika, Ilam, got it? Martin,
Yes, sir.
Got it.
right? So you can take the screenshot and you can draw it and write down whatever written in like my handwriting. So we'll keep till this point. But draw it in your notebook because you will require it. The most frequently asked question is draw the well-level diagram of non-cyclic photophosphorylation. Photosystem second. Difference between dark and light reaction. These are the question form. You will get these, encounter these questions in your school exam. Clear?
Yes, sir.
You will draw it after the class. Even you can draw it right now if you want. It's simple. Draw it right now, yeah. Don't leave it for after the class. Draw it right now. It's easy. You have not to draw everything. Whatever in circle that you have to draw it.